The topic of this podcast is glycolysis. Glycolysis literally means the splitting of glucose, the 6-carbon sugar that is so important to the metabolism of cells. The enzymatic process of glycolysis occurs in the cytoplasm and is the starting point for cellular respiration. Almost every cell uses glycolysis regardless of the type of cellular respiration they are able to carry out. Because of this, we believe that glycolysis probably arose very early in the evolution of life on the planet. In glycolysis, glucose is partially broken down in 3 stages. The end product of this breakdown is pyruvate. You'll get 2 of these 3 carbon molecules of pyruvate or pyruvic acid from a single 6-carbon glucose. In the process, glycolysis produces 4 ATP for a net gain of 2 ATP, as well as 2 molecules of NADH. Now we'll discuss the 3 stages of glycolysis, which leads to these results. But first, you should recall that ATP is the important energy storage molecule for a cell, and many reactions in the cell require the input of ATP. Thus, the cell needs to constantly restore ATP levels. ATP stands for adenosine triphosphate, a 3-phosphate molecule. It's usually converted to adenosine diphosphate, the 2-phosphate form, in reactions that remove 1 phosphate. The first stage of glycolysis is investment. During this stage of glycolysis, the glucose molecule acquires 2 phosphate groups. By acquiring a phosphate, the sugar is trapped within the cell and can't cross the membrane, and then it acquires a second phosphate group. The phosphates are donated by ATP molecules, which in turn are converted to ADP. After this event, the sugar is split into 2 3-carbon molecules, each with a phosphate. So in summary, during the investment, 2 ATPs are used to transfer phosphates to the sugar and thus are converted to 2 ADP molecules. 2 3-carbon molecules result at the end of this stage, each containing one of the phosphates taken from ATP. During the second stage, an important redox or oxidation reduction reaction occurs. But first, one enzyme converts one of the 3-carbon molecules that resulted from the splitting of the 6-carbon sugar into another form. This gives you 2 of the same 3-carbon molecules with the phosphate, called G3P. Next, an enzyme catalyzes a double event. First, the 3-carbon molecule is oxidized and NAD+ is reduced to NADH. Simultaneously, a free phosphate group from the cytoplasm in the cell is used and added to the 3-carbon molecule. This happens to both of the 3-carbon molecules made from the first stage, so 2 phosphates from the cytoplasm are used and 2 NADHs are produced. Each NADH is carrying 2 energy-rich electrons away from the glucose, and these electrons can be used by the cell to do work. The use of these electrons from NADH will be discussed in a later lecture on the electron transport chains in the mitochondria. Importantly, at the end of the second stage, we have 2 3-carbon molecules with 2 phosphates each, and we have produced 2 NADH molecules from the 2 NAD+ molecules. In the final harvesting state of glycolysis, all 4 of the phosphates present on the 3-carbon molecules will be transferred to ADP molecules to produce ATP. Because both of these 3-carbon molecules can donate 2 phosphates to 2 ADP molecules to make 2 ATP molecules, the sum total of ATP made will be 4. 1, 2, 3, 4. This means that the cell will be ahead or will have gained 2 ATP molecules per glucose since we initially invested 2. The 3 carbon molecules left at the end of these reactions is called pyruvate. There will be 2 pyruvates made per glucose. Remember, all of the molecular conversions and transfers occur because of enzymes. In summary, the process of glycolysis invests 2 ATP and 2 NAD+ molecules to split glucose. The end result will be 2 pyruvates, 2 NADH, and 4 ATP, or a gain of 2 ATP. I hope this podcast helped clarify this important molecular process in cells. See you next time.